right, you guys, welcome back. Here we are with chapter 11 again, gene expression and epigenetics. Today we're going to be looking at our section 2 now, so control of gene expression. Now in our last section of notes, we were talking about what epigenetics is, how it is a change in the expression of genes based on not so much the actual DNA bases, but more so on these chemical groups that associate with the DNA. So epigenetics itself means outside the cell or outside the genes, meaning that something else has to occur that's going to cause a change to these chemical groups, which will change what genes the transcription factors are able to access or not access. So let's move my little screen a little bit higher. Remember? All right. And then, there we go. So control of gene expression. Combination of signals instructs cells to activate combinations of transcription factors, and these in turn control which genes are being transcribed. So if you remember the central dogma of molecular biology, DNA codes for the RNA and the RNA codes for the protein. So you have to take the DNA, you have to transcribe it into the messenger RNA, and that takes the information from a gene in the DNA out of the nucleus and into the cytoplasm, where it's picked up by a ribosome, and there's the tRNAs with the amino acids, and we translate the message in the mRNA into an amino acid chain, which then gets folded up to make a protein. So let's talk a little bit about what that means in terms of what this is saying. How is it that we know when to transcribe genes into the mRNA in the first place? We've mentioned before that there are transcription factors, and what these do is they're the ones that target specific genes. Like, all right, we are going to look at this section, we are going to transcribe to this point, and that's it. Now, there are multiple transcription factors, and the actual combination of transcription factors will affect which gene gets transcribed. Because remember, most of your DNA actually isn't encoding for proteins. Only about 1.5 to 2% encode for protein. So the transcription factors, different combinations are going to look at the DNA and transcribe portions of it and process portions of it in order to get particular proteins. And there are different signals that tell the cell when to do it, how often to do it. Should you keep it on all the time? Should you just keep transcribing this gene over and over and over? Or should you do it for a limited time and then stop and then possibly start up? Or are you going to be, silenced forever? So a whole bunch of different signals decide all of that. When to do it, how often to do it, how quickly to do it. And then the transcription factors determine the gene that is going to be transcribed and thus the protein that is going to be made. So there's a lot going on in this. I mean, each different combination of signals and transcription factors is going to lead to a different result. There's too many for us to talk about in this class, but it's just kind of crazy how complex it is, and also kind of cool if you ask me. All right, so chromatin remodeling. Now, in the last section, we mentioned that there are these chemical groups that associate with the DNA, and changes to these chemical groups can affect whether or not genes get transcribed. So again, here's our DNA. Now what happens is these groups are actually on the histone, but we like to say they associate with the DNA because the DNA is wrapped around the histone. So if you look at the picture, this circular glob right there, that is a histone. It's a particular protein that the DNA wraps around in order to condense itself so that it fits nice snug in the nucleus. And whether we add groups or remove groups, that's going to affect how those genes get expressed. Now, there are three major ones we're going to talk about, and we're going to talk a little bit about especially how two of them affect the transcription and all that. Now, as we also mentioned in the last section, your body naturally turns genes on and off as you grow and develop. When you are an embryo, the gene for the embryonic hemoglobin is turned on, and is being expressed, while the ones for fetal and adult hemoglobin are turned off, so they are not expressed. Then you develop into a fetus, the embryonic one turns off, fetal one turns on, adult hemoglobin stays off. And then you're born, and then the embryonic one is still off, the fetal one turns off, and the adult hemoglobin gene turns on and remains on. So these groups, they're not something necessarily completely new to your DNA. You already have them on your DNA, helping to control the process of gene expression. But adding them or removing them could affect the expression of certain genes, which can lead to certain traits, disorders, etc. We'll talk more about those in a little bit. All right. So again, there are three main groups that we are going to talk about. These bind to the histones and will affect how the DNA wraps around the histones. So the three that we're going to talk mostly about are acetyl groups, methyl groups, and phosphate groups. Phosphate groups, more so good to mention, but we're not really going to get into them. They kind of do a little bit of both of the jobs that acetyl and methyl do, but not necessarily to the same degree. And also, it depends on the circumstances. Acetyl and methyl groups are the ones we're going to focus the most on. Now, acetyl groups, these are the ones that when they are added to a gene or to a histone, they affect it and allow a gene to be transcribed. So if the gene is being transcribed, that means it's going to be expressed. And methyl groups do the opposite. They actually prevent gene expression. We will talk a little bit about those coming up, but I at least wanted to introduce that idea to you now. So acetyl allows for transcription and methyl prevents it. Again, phosphate kind of does a little bit its own thing, kind of does a little bit of both depending on the different circumstances. Sorry about the light coming in and out. It's going in between sunny and cloudy all day today. All right, so let's see. So as we talked about the histones, these are very, very important proteins. The DNA wraps around them and then allows them to be either transcribed or not transcribed, so expressed or not expressed. And how it kind of works is if you kind of look at this picture here. So here we have on the top, you notice all these little M's. That means that there are methyl groups added. So if you notice, it's all kind of blocked off. Nothing is being expressed. But when the acetyl groups start getting added, more and more acetyl groups, they unwind and it allows the DNA to be expressed. We're going to talk a little bit more on the next slide, because if you notice, we got a little glob there that says acetylation. But it's because of the histones and how these chemical groups attach to them will actually affect the histones, causing it to shield the DNA, thus not allowing transcription and gene expression to occur, or exposing the DNA to allow transcription and gene expression. Let's take a look at acetylation. So acetylation is when you have acetyl groups added to the histone. Now, it says nucleosome controls gene expression through the acetylation of specific amino acids on specific histone proteins. Let's do a little review. So we said we have the DNA, and then it wraps around the histone. Now, as we see in this picture, there are multiple histones there, and the DNA is wrapped around the histones. And then when all of these DNA histone complexes kind of wrap and squeeze together, that's what a nucleosome is. It is multiple histones with DNA wrapped around them, all kind of coiled up together. So because we have the DNA not just being wrapped around histones, but also being these histones wrapping up and coiling together, that's why we say the nucleosome controls the gene expression. Now, when we add acetyl groups, it's actually causes the histone to contort and change its shape. So it kind of modifies and twists the histone so that the DNA becomes more accessible. So it's exposing more of the DNA by bending and contorting the histone. And that's all because of how we bind this group to it. The binding of the acetyl group to the histone causes the histone to change shape. When it changes shape, it kind of loosens up the DNA. And now that the DNA is loosened up, the transcription factors can get in there and get to that gene and can now transcribe it. Now, if you take the acetyl group off, it'll go back to its normal contortion, which means that's going to be its normal twisted strength on the particular gene. So different transcription factors may or may not be able to get to it. When the acetyl group is attached to it, it's going to be very, very easy for the transcription factors to have access. So that gene is pretty much going to be on as long as that acetyl group is on, meaning it's going to keep getting transcribed so long as that acetyl group is present and contorting that histone to allow for the transcription factors to enter. All right. RNA interference, or RNA lylae. Right there, see? RNAi. Now, remember how we said most RNAs are single-stranded? This is true. But RNAis are actually one of the only double-stranded RNA molecules. And the whole point of them is to bind with mRNAs, and once they bind with the mRNA, it marks them for destruction. So we have our DNA, we transcribe it to make this red RNA here. That's the messenger RNA. But if the RNAi binds to the mRNA, so the RNAi starts out single-stranded, but then it binds to the mRNA, causing it to be double-stranded. Double-stranded RNA automatically gets destroyed. Now, since it's being destroyed and you can no longer have the anticodons of the tRNA line up with it because, hey, look, it's already got buddies, it can't make the protein. So that's one way that it can affect protein expression. But another way is what this RNAi, it doesn't just mark mRNA for destruction. It also can add methyl groups to the histones, and or this one can be to the DNA or to the histones. And this occurs when it's still in the nucleus, so it's not like it's out in the cytoplasm at this point, but it can actually add methyl groups to the DNA. And when they add the methyl groups to the DNA, it has the opposite effect as the acetyl groups. Remember, the acetyl groups bonded or attached to the histone, and that caused the histone to contort and loosen up the DNA so the transcription factors can have access to it. When the methyl groups bond to the DNA or bond to the histones, it actually causes everything to tighten up. So it tightens and twists up even more so than it was before. Now that everything is tighter, it means it's extra hard. The transcription factors cannot access it at all. So that is going to prevent the transcription factors from accessing it, which means there's not going to be any transcription of that particular gene. So preventing transcription means it's not going to be expressed. And so long as that methyl group is there, it will stay that way. Now, on the other hand, if the methyl group is taken off, it loosens it up to its normal winding, and transcription factors may or may not be able to access it depending on the combination of transcription factors. But again, when the methyl group is on, it twists it, causes it to contort and twist up on itself. So it's going to further prevent any access to that gene, thus or preventing gene expression, or as we say, silencing the gene. Because if it can't be expressed, you don't see what it's supposed to be doing, which means it's essentially as if it's not even there. All right, and that looks like that is the end of section 2. So in conclusion, as a reminder, these chemical groups can be added to the DNA or the histones, and they will affect DNA expression. The 3 main groups are acetyl groups, which allow for transcription, methyl groups, which prevent transcription, and phosphate, which do a little bit of both. Now, how this works, acetyl groups bond to the histone, causing it to contort and loosens up the DNA, so the transcription factors have full access to that gene. On the other hand, methyl groups, when you add them to the DNA or the histone, it causes everything to tighten up even more. So now no transcription factors can have access, thus silencing the gene from being expressed. So I hope that helped. I hope it kind of made some of the material a little more understandable. If you do have any questions, definitely remember, hit me up in our Google Classroom or email me anyway, or talk to me in class if I ever see you guys in class again. I know it's kind of that COVID-19 gunk going on right now, but still, if you have any questions, I'm here for you. Please feel free to ask. The link for this Prezi will be in the description in YouTube, but until then, I hope you guys stay safe, stay well, and I'll talk to you later. All right, bye-bye.